Welcome back, students. Today we are going to discuss part 10 of our unit 2, that is unit of life. Okay? But before start the made content, I would like to inform that the images used in these slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT or have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. And in today's video lecture, mainly we are going to discuss the process of meiosis. In the last lecture, we already discussed about cell reproduction or cell cycle and mitosis. Okay, so in today's video lecture, we are going to discuss this meiosis part. Now, what is meiosis? Meiosis means production of offspring by sexual reproduction. Okay, so process if we produce offspring by sexual mean, then fusion of two gametes is required, and each gamete contains a haploid set of chromosome. Okay, so for the through meiosis this haploid set of chromosome has been produced. Okay, so the production of offspring by sexual reproduction includes the fusion of two gametes, egg and sperm cells, having haploid cell of chromosomes. So the gametes are formed from the specialized diploid cell. Okay, and this specialized kind of cell division that reduce the chromosome number by half as a result in the production of haploid daughter cell is called meiosis. Okay, so this is the definition. What is the definition? What kind of division is this? Meiosis is specialized type of cell division by which reduction of chromosome number just half by just half has been occur. Okay, so from diploid cell to haploid cell, this kind of cell division is known as meiosis. So can you write this down? Specialized kind of cell division, this is a specialized kind of cell division that reduce the chromosome number by half as in the production of haploid diploid cell. Okay, and meiosis actually ensure the production of haploid phase in the life cycle of sexually producing organism, where fertilization again restore its diploid phase. Okay, fertilization occur between ova and sperm, right? So both contains n number of chromosome, and after fertilization it will again restore its original number of chromosome, that is 2n. Okay, so through meiosis, we can produce haploid cell in which half number of chromosome is present. Okay, in the life cycle in sexually reproducible organism, and after fertilization, it again restore its diploid phase. Okay, so we come across meiosis during gametogenesis in plants and in animals. Okay, and this meiosis leads to the formation of haploid gamete. Formation of haploid gamete. Formation of haploid gametes occur through meiosis. Okay, so this is meiosis. So what are the key features of meiosis? Let's discuss the key features of meiosis. Meiosis involves two sequential cycle of nuclear and cell division. Okay, as you know this in mitosis only one cycle is there. Okay, one cycle of nuclear division means karyokinesis and cytokinesis, while in meiosis two sequential cycle of nuclear and cell division called meiosis 1 and meiosis 2. Okay, but only single cycle of DNA replication means DNA only replicate in S phase. There is no duplication of DNA occur again. Okay, so only single cycle of DNA replication occur, but two sequential cycle of nuclear division and cell division, means meiosis 1 and meiosis 2. Right. So this meiosis 1 is initiated after the parental chromosome have replicate to produce identical sister chromatids at S phase. Okay, and meiosis involve pairing of homologous chromosome and recombination between non-sister chromatids. Recombination between non-sister chromatids of homologous chromosome, right? Of homologous chromosome. Just clear this thing. Okay, so meiosis involve pairing of homologous chromosome, but recombination occur between non-sister chromatid of homologous chromosome. And after, what is the result of meiosis? At the end, we will get 4 haploid cells at the end of meiosis 2. Okay, so first point, meiosis involve two sequential cycle of nuclear and cell division known as meiosis 1 and meiosis 2, but only single cycle of DNA replication. Okay, number 2, meiosis 1 is initiated after parental chromosome have replicated their DNA or identical sister chromatids at S phase, and meiosis involve pairing of homologous chromosome and recombination between non-sister chromatid of homologous chromosome. And at the end of the meiosis, we will get 4 haploid cell at the end of meiosis 2. Clear? So we can divide all the meiosis 1, all the events which are present in meiosis 1 is exactly same to meiosis 2, but in prophase 1 there is a difference, as in prophase 1 recombination or crossing over occur in this prophase 1. So this one is a longer phase. Okay, so we can further divide meiosis 1 and meiosis 2 into 4 different subcategory: prophase 1, metaphase 1, anaphase 1, telophase 1. Similarly here, prophase 2, metaphase 2, anaphase 2 and telophase 2. Okay, now these are the stages of meiosis 1, as you can see over here. This picture I have taken from your NCERT book. So this is the prophase 1. Okay, you can see crossing over, X chiasma formation or recombination, right? So recombination occur during this phase, crossing over occur in meiosis 1. And metaphase 1, it is just similar like mitosis, all the chromatids just align on the equatorial plane. Okay, and ester rays bind with the kinetochore. Okay, and this anaphase 1, separation of homologous chromosomes occur. Okay, and then telophase 1, one cell to two cell. Okay, and then immediately that both the cells again start nuclear division, and then metaphase 2, anaphase 2 and telophase 2. So 1 cell to 4 cell, 4 haploid cell. Now let's discuss all the events one by one. Okay, so in meiosis 1, this is the prophase 1. Okay, so prophase of the first meiotic division is typically longer and more complex if we compare this prophase with mitosis, because there is no crossing over, no recombination occur during mitosis. Just that's why the progeny or offspring cells are just show their identical, exactly identical to their parent cell. Okay, so equal division uh, in mitosis, equal division of chromosome occur there, but and no crossing over, no chiasma formation, no recombination, nothing like this. So all those things are the peculiar characteristic of prophase 1 of meiosis 1. That's why it is typically longer and complex if we compare this with the prophase of mitosis. Okay, we can further divide prophase 1 into 5 different phases, and it depends upon the on the basis of what? On the basis of chromosomal behavior. On the basis of chromosomal behavior, these 5 phases are: first one is leptotene, then zygote, okay, then pakitine, then diplotene, and the last one is dikinesis. Okay, so prophase 1 is comparatively longer and complex. We will further divide this prophase 1 into 5 different phases: leptotene, zygote, pakitine, diplotene, and dikinesis. Okay, so now we will see the behavior of chromosome in each stage. It will behave differently. Like during leptotene stage, chromosomes become gradually visible under light microscope. Normally, they are present in the nucleus in a beaded-like structure, chromatin form. Okay, but they start condensing itself, so they become gradually visible and compact itself. Okay, and continue compaction is continued throughout the leptotene. Okay, so what is the main point of this leptotene stage is chromosome become gradually visible under light microscope, right? As you can see over here, this is the uh, prophase 1, and just now we have discussed that prophase 1 we will further divide into subcategories, 5 subcategories. So this is the leptotene. Okay, here there is chromatin start condensing, and we can now able to see the leptotene. This is zygote stage. Second stage is zygote, in which chromosomes start pairing up, means bivalent. You can see bivalent. Okay, how? Because of the presence of synaptonymal complex. Okay, so this is zygote, then pakitine, then diplotene, and then dikinesis. These are the 5 sub phase of prophase 1, right? So after leptotene, the second stage is zygote. Second stage is zygote. So during this stage, chromosomes start pairing together, and this pairing up is known as synapses. So they start forming pairing up, and at this time we can use the term homologous chromosome. Such pair chromosome, what is the correct term for that? Homologous chromosome. Okay, and electron micrograph of this stage indicate the chromosome synapses accompanied by the formation of a complex structure known as synaptonymal complex. Okay, so because of the presence of synaptonymal complex, uh, chromosome start pairing up, okay, bind to each other, and right now they are showing homologous chromosome. This is the characteristic of zygote stage. So synapses, they start pairing up, called synapses. And how? Because of the formation of synaptonymal complex. And this time we can use the term bivalent for these homologous chromosomes, like this one, and oh, just a second. Because of this synaptonymal complex. This time we can use the term bivalent. Okay. However, this bivalent form is more clearly visible into the next phase of prophase 1, and this form is relatively short. Okay, so next stage is pakitine. So in pakitine, 4 chromatids of each bivalent chromosome become distinct and clearly appear as tetrad. So this is the characteristic feature of pakitin. So in zygote, they looked like this. Okay. And right now we are using the term bivalent for the same. These are the homologous chromosome. Homologous chromosome. And as it enter into the pakitin stage, they separate out their sister chromatids like this. So these two are sister chromatid and these two are sister chromatid, right? And right now we are using the term for this paired group of homologous as tetrad. Okay, so this tetrad is clearly appear in pakitin stage and as a bivalent zygote. Is it clear? So in leptodine, chromosomes start condensing themselves, and in zygote they start pairing up as a bivalent, and these right now we are using the term for them homologous chromosome. Okay, because of the presence of synaptonemal complex. So synapses formation. Okay, synaptonemal complex, and here we can use the term for this homologous chromosome bivalent. And as it enter into the pakitin stage, they separate out their chromatids. Okay, they are chromatids. So four chromatids we can see over here. Four chromatids. This one first, second of one chromosome, and three, four, second chromosome. These both are homologous chromosome. Okay, so right now we can say one, two, three, four. So four chromatids we can see in pakitin stage. So that's why we can use the term tetrad during pakitin. That's why it is written over here. During pakitin, the four chromatids of each bivalent chromosome become distinct and clearly appear as tetrad. Okay, this stage is characterized by the appearance of recombination nodules. Means, what is this recombination nodules? Those sites at which crossing over occur between non-sister homologous chromosome. Okay, non these both are homologous chromosome, but this chromatid is behave like a non-sister for this chromatid. These both are uh, sister chromatid. 3 and 4 and 1 and 2 sister chromatid. Okay, so crossing over occur between non-sister chromatid, means 1 and 2 are sister chromatid, 3 and 4 are sister chromatid, but crossing over occur between 2 and 3 which are non-sister chromatid. Okay, okay, I can just draw some line. I just draw some line just to for your clarification. Okay. So these are homologous chromosome. Now again, these are homologous chromosome. They start separating their chromatid. Okay, so 1 and 2 are act as a sister chromatid, 3 and 4 act as a sister chromatid, but crossing over occur between non-sister chromatids. So what is the situation right now? Situation is like this. Hmm, I must draw it again. Okay. Situation must be like this. So 1, 2 sister chromatid and this 3 and 4 non-sister chromatid. So crossing over occur between 3 and 4. 3 and 2 non-sister chromatid. These are non-sister chromatid. Okay, so crossing over is the exchange of genetic material between two homologous chromosomes between non-sister chromatid of two homologous chromosomes. This is again a peculiar character of Pachytene. So what is the first character? They start looking at as a tetrad. This one is the first stage, first peculiar character. Second, crossing over occur during Pachytene stage or because means exchange of genetic material between non-sister chromatid of homologous chromosome. Okay, and this crossing over is a enzyme mediated process, and the enzyme involved in this process is known as recombinase enzyme. Recombinase enzyme. Okay, clear? So, crossing over leads to recombination of genetic material of two chromosomes. Recombination between homologous chromosome is complete by the end of Pachytene. Okay, leaving the chromosome linked at the site of crossing over. Clear? Okay. So, after Pachytene, diplotin. Next phase, diplotin. The beginning of diplotin is recognized by the dissolution of synaptonymal complex. Now, that pairing occur because of the presence of synaptonymal complex, right? So this synaptonymal complex start dissolving itself. So tendency of homologous chromosome of bivalent to get separate out from each other, except at the site of crossing over. Except at the site of crossover. So can I write down? Can I draw a diagram again just for you? Okay, this is the 1 and 2 and they start separate out to each other. Okay, they start separate out and this is the 3 and 4. These are the homologous chromosome, right? Because this, why they start to separate out? Because this synaptonymal complex is start dissolving itself. Okay, so these bivalent homologous chromosome of the bivalent to get separate out except at the site of crossover. Crossover इस तरीके से बना रहेगा and that's why we can now use the term chiasma. This X shape structure, X shape, right? Again, X shape structure is known as chiasma. Now, in diplotin, we can see that stage of crossing over as a chiasmata. Okay, as a chiasmata. Now, just see this diagram. So, this beaded structure start condensing and form chromosomes in zygotine, bivalent formation, right? Means two homologous chromosome come closer because of the presence of synapses, synaptonymal complex. In Pachytene, crossing over starts. Okay, and at this time, both the chromatids separate out of homologous chromosome and crossing over also occur during Pachytene stage. Means exchange of genetic material of non-homologous chromosome, non-homologous, sorry, non-sister chromatid of homologous chromosome in Pachytene, but we can see this crossover in diplotin when bivalent starts separate out and only attached towards the at that particular location where crossing over occur. See, as you can see over here, right here we can see the exchange of material. So chiasmata formation is the character of diplotin. Okay, and but crossing over is the peculiar character of pachytene. Okay, what is the peculiar character of zygotane? Bivalent formation. Bivalent. Okay, in pachytene, tetrad is formed, means sister chromatid separated out. Okay, so these are the main events occur at each sub phase of prophase 1. Clear? Okay. Now, what is the final stage of meiosis 1 of prophase 1 is dikinesis. Okay. And in dikinesis, this is marked as a terminalization of chiasmata. In dikinesis, terminalization of chiasmata occur, means separation of that position where crossing over also occur, gets separated out. So terminalization of chiasmata. So during this phase, the chromosome are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. Okay, spindle fully condensed and the meiotic spindle is assembled to prepare homologous chromosome to get separate out. By the end of dikinesis, the nucleolus disappear and nuclear membrane also broken down. This is the characteristic feature of prophase, right? Nucleolus will start disappear, nuclear membrane also broken down, and then after that, that prophase stage of the cell is in that prophase 1 stage come into the metaphase stage, right? So dikinesis represent transition to metaphase. Okay, so terminalization of chiasmata is its peculiar character. Okay, and nucleolus disappear, nucleus membrane also get disappear in this particular stage. After prophase 1, cell come under metaphase 1 stage. So here the bivalent chromosome align on the equatorial plane, as you can see in the picture. Okay, and the microtubules from opposite pole of the spindle attach to the kinetochore of homologous chromosome. Okay. Now, in anaphase 1, the homologous chromosomes separate while sister chromatids remains associated with their at their centromere. Means only homologous chromosome gets separate. Okay, and in telophase again, this is just a reverse of prophase 1. So nuclear membrane and nucleolus reappear, cytokinesis start, and this is called dyad of a cell. Okay, so like this. Nuclear membrane again start appear, nucleus also start, and separation of cytokinesis start. Okay, and this time we can use the term dyad of a cell. Clear? Now, although in many cases the chromosome do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between two meiotic division is called interkinesis. And this interkinesis is generally short-lived, and in during interkinesis there is no replication of DNA occur. Replication of DNA occur in interphase, that is a um, phase of cell cycle. If I can draw this kind of a structure, this one is G1 S G2. So DNA replication occur in this phase only, DNA replication, and this M phase or mitosis or meiosis, what M phase. This whole is known as interphase. So please don't confuse interphase with interkinesis. This interkinesis is a part of mitosis. Okay, so there is no replication of DNA occur during interphase. Replication of DNA occur only in S phase of interphase of cell cycle. Clear? Okay. So this interkinesis is followed by prophase 2, which is comparatively simpler than prophase 1. because now there is no crossing over, no chiasmata formation occur during prophase 2. So in meiosis 2, prophase 2, meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes has fully elongated. In contrast to meiosis 1, meiosis 2 resemble to a normal mitosis, means immediately nuclear membrane again start disappear at the end of prophase 2, nucleus also start disappear, and chromosome again start compacting. Okay, chromosome become compact again in prophase 2, and at metaphase stage all the chromosome align on the equatorial plane, and the microtubules from the opposite pole of the spindle get attached to the kinetochore of sister chromatid. Okay, kinetochore is a plate like structure which is present on, if this is centromere, this kind of plate, this is known as kinetochore. Kinetochore. Okay, so spindle fiber attached to this kinetochore of sister chromatid. Right. Okay, these are the stages of meiosis 2. Homologous chromosome already separate out during meiosis 1. Okay, now they again start condensing to each and showing in a compact form, and in metaphase 2 they are aligned on equatorial plane. Okay, and spindle fiber attached to the kinetochore of sister chromatids in both the cells. And anaphase 2, they microfibrils shortening of microfiber occur, so sister chromatid get separated out, as you can see in this particular picture. And telophase 1, they get separate out, and haploid number from one cell to 4 haploid numbers of cell you can get, as again nuclear membrane start appear. Okay, nucleolus start appear. Okay, so these are the stages, and all the stages of meiosis 2 like prophase, metaphase, anaphase, telophase, these are like just similar to mitosis. Okay, anaphase 2, it begin with the simultaneous splitting of centromere of each chromosome. Okay, which actually used to hold the sister chromatid, means sister chromatid gets separate out during anaphase, allowing them to separate towards opposite pole. And how? By shortening of microtubule attached to the kinetochore. Clear? And in telophase 2, meiosis ends with telophase 2, in which two groups of chromosomes once again get enclosed by nuclear envelope, and then cytokinesis followed, the result the formation of a tetrad of a cell, means 4 haploid daughter cell. Clear? So what is the significance of meiosis? Meiosis is a mechanism by which conservation of specific chromosome number of each species is achieved across generation of sexually reproducing organism. Okay, it also increases genetic variability because exchange of genetic material occur. Okay, so that's why meiosis increase genetic variability in a population of organisms from one generation to another generation. Variation are, as you know, this variation are very important. These are very important for the process of evolution. So this is the significance. Now, if we compare mitosis with meiosis, then number of chromosome in the daughter cell is 2n diploid. It is in haploid number 1n. These are the clones. All the offsprings are the clones of their parents. While meiosis, daughter cell is different from their parent cell and from each other too. Okay, some genetic information in parent cell, same type of genetic information because they are just clone. Okay, while in daughter cell have half number of chromosome as somatic cell. Okay, so this is the shuffling of genes occur. in meiosis, while it is not happened in mitosis. So this is all about the meiosis and their steps. The main information or main important question comes from the prophase, leptotin, zygotin, packaging, diplotin, dikinesis stage. So you have to remember what is the main event occur in each phase of prophase 1. Okay, still you have any query, any doubt, you can write down to me, comment box, we will discuss that query later on. Till then, take care, bye bye, namaskar.